So today, 1st October 2020, it's my pleasure to introduce Eva Maria Krämer Albers from Germany. Eva Maria received a PhD in molecular and cellular neuroscience from the University of Heidelberg, Germany, and was further trained in neurogenetics at the Max Planck Institute of Experimental Medicine in Göttingen in Germany. So she got fascinated by EV in 2006 when she discovered the release of exosomes by myelinating oligodendrocytes. Eva Maria is heading a group at the Johannes Gutenberg University in Mainz, Germany, studying the role of EVs in neuron-glia communication and long-term CNS integrity. And furthermore, EVs released in the circulation during exercise. She served as associate editor for the Journal of Extracellular Vesicles between 2012 and 2019, and is vice president of the German Society of Extracellular Vesicles of GCEF. But today, Eva's lecture is about transfer of exosomes from myelinating glia to neurons, the implication for neural support and long-term neuronal maintenance. Welcome, Eva. Please go ahead.
Thank you very much, Carolina, for the kind introduction. Okay, let's start. Again, thank you, Carolina, for the kind introduction. And our research group is focused on the physiological function or the physiological function of EVs in neural cell communication. And so we would like to study what is going on in our brain every day, and probably as we sit here, how are EVs exchanged between neurons and glial cells, and what is their physiological function. So as you can see here, neurons, they are really complicated cells. They have a really complex architecture. You can see a group of cell bodies here sitting together, and they extend long processes, and these are the axons. They are very thin, long processes, and so these need to be maintained over our whole life. And I would like to keep that in mind during the whole presentation, that neurons need to be maintained in our brain over our whole lifetime. So the presentation will be divided in largely 3 parts. First of all, I will introduce you to our results that we have largely published already, and the analysis of this oligodendrocyte to neuron EV transfer, which is largely based on cell culture data, in vitro data. So in the first part, I will give you the basis, our cell culture basis, where we analyze the oligodendrocyte to neuron transfer of EVs. This is largely published. And in the second part, this is what kept us busy over the past few years, where we tried to validate these data actually in vivo to see whether what we found in vitro really also happens in the brain. And then in the third part, I will go more into the functional implications regarding long-term neuronal maintenance, and they're also from the in vivo perspective, looking largely at transgenic animals. So when thinking about the cellular architecture of the brain, most people largely think about neurons and the networks that these neurons build, and forget about the glial cells. But glial cells are very important items in the brain. And also the interactions between the glial cells and the neurons, they keep actually the whole network in place. They are important for the development of the network, for the plasticity of the network, and also for its maintenance. And there are different cell types, and we are focusing on oligodendrocytes, which are the myelinating cells of the CNS, and they insheath axons and insulate them. And this insulation results in the fast propagation of action potential, which is called saltatory conduction. But on top of this, they also provide support to axons. And this has been recognized in the past when people started to do knockout animals. So in the late 90s, the major myelin protein, which is called PLP, has been knocked out. And people were very surprised to see that myelin in general, here you can see it lying on an axon, this is a longitudinal section, is still intact. Also a second knockout, the CNP, also a myelin protein, the myelin sheath is still intact, but the axon develops swellings and it starts to degenerate. So these are glia-specific genes, and if they lack, the axons degenerate. It's not the oligodendrocyte that suffer, but the axons. And this is how people discovered the support function of oligodendrocytes. And if this support is lacking, axons slowly start to degenerate. And since then, it has been studied what is really the molecular basis of this support, and there is some progress, but it's still not completely understood. And of course, this is very relevant for myelin disease, where when myelin is degenerating or degraded or any other defect in oligodendrocyte occurs, then what very often is observed is axon loss. And this is actually what the people really make sick and what is finally irreversible in a disease. So therefore, it's very important to understand the basis of this trophic support. So when looking at the whole brain, then we have learned in the past years that all cell types actually release extracellular vesicles, neurons and the glia cells. And these vesicles travel inside the brain and address target cells. And some functions has been added to these, largely based on tissue culture data. And it's very difficult to study this in vivo, since the brain is a very compact tissue, and this has largely to be addressed by transgenic animal studies. And this is what has to be brought forward also regarding EV transfer and understanding EVs in the brain. We are focusing on oligodendrocyte neuron interaction during this talk and the role that these EVs play. So we characterized oligodendroglial EVs quite extensively, I have to say, by biochemical means, but also by morphological means. And what we find, we use different types of isolation methods. Largely, we are analyzing 100 K pellets, because we find that they very reproducibly carry small EV and exosomal markers, but also using other isolation methods. And only a few markers are shown here. We are looking at many more, and we also find ILIX and TSG101, so the typical endosomal markers. And we think that these largely reflect exosomes coming from endosomal pools. And we also perform proteomics, but the idea that this reflects exosomes largely comes from the observation that, that we find a strong enrichment of multivesicular bodies in primary cultured oligodendrocytes. And we also find small vesicles very reminiscent of exosomes, because they are very homogeneous in shape outside of the cells. And maybe this could be a fusion profile. So we are quite convinced that at least substantial fractions of what we isolate in our 100K pellets, or in 10, or what is represented in 10K supernatants, is actually reflecting exosomes. And this is further supported by the finding that when we look in situ, in cross-section of axon myelin units, we also find these multivesicular endosomes, and in particular prevalent in places directly opposed to the axon. So this is the inner oligodendroglial cytoplasmic loop. You can see the myelin layers on top, and this is the axon inside, and this multivesicular body is direct in the apposition to the axon in the periaxonal space. Here, and we find this very prevalent. We can see these figures very, very often, which is actually surprising for electron microscopists. And this actually inspired us to think that exosomes might play a role in axon-glia communication, which is very important to keep this whole axon myelin unit intact over its whole life. And as I said, the MBBs in these adaxonal spaces, they are highly prevalent, and we see them very often. So we studied exosome secretion and their role. And just to summarize, I show you a few of these data, but just to give you the main results, we found that the secretion of exosomes from oligodendrocytes and from these multivesicular bodies is actually triggered by neurotransmitters, such as glutamate. That neurons internalize these exosomes by endocytosis, and that the neurons that receive these exosomes are more stress resistant. Now, this is all based on in vitro studies that I'm now summarizing in the next few slides. We largely use the Transwell co-culture system, where we grow oligodendrocytes on filter membranes and neurons in the bottom well. So all is based actually on primary cultures. And then we can manipulate this system. And if we treat oligodendrocytes with glutamate directly, we can see a strong increase of small EV or exosome release. PLP here is the main myelin protein, which can also be used as a kind of a marker of these oligodendrocyte-derived EVs. And this actually depends on glutamatergic activity of the neurons that are growing below these oligodendrocytes. If these are stimulated by pharmacological means to fire action potentials, and in particular to release glutamate, then we can see an increased release from oligodendrocytes that are growing in the top well. And if these oligodendrocytes are deficient in the NMDA receptor subunit NR1, then this increase is not present anymore. So this depends on oligodendroglial glutamate receptors of the NMDA receptor subtype. And neurons that are stained, and if the oligodendrocytes in the top well are either stained with pKa67 or also transfected with an, let's say, PLP-EGFP, then we can find these EVs inside the neurons that are grown in the bottom well. And they seem to be accumulated in endosomal compartments. So one important question was whether these exosomes or small EVs that are delivered to the neurons, whether the cargo is actually retrieved or degraded. So in the endosomal system, it may well be that cargo degradation occurs and there is not much of a functional implication of this cargo. So to test for this, we used Cre recombinase as a reporter, which we loaded into these exosomes by viral overexpression. And then we're growing these manipulated oligodendrocytes together with neurons carrying a reporter gene. And if the exosomes are then transferred and the cargo is actually retrieved and functional, and this is an important point of the Cre system that we always have to keep in mind. So there, the cargo really has to be functional, and it has to be also be delivered to the nucleus and then activate the reporter gene. So, and indeed, what we see is that CRE was recovered and the reporter gene is activated in target neurons. And this is actually also the case if we inject these CRE manipulated exosomes into the brain of reporter mice that carry the reporter gene in any cell of the body. So there, when we inject into the hippocampus, we also see recombined neurons, which for us was a very important proof of principle experiment that this actually, at least the uptake and the cargo retrieval also can occur in vivo. But again, what you should keep in mind, this is a viral overexpression. CRE is a nuclear protein, and I think per se, it is not given that CRE is actually, the nuclear version of CRE is actually sorted to an extracellular vesicles. And we definitely had to overexpress CRE to get it into the EVs in our case. One other experiments we performed, the question was, are these EVs then taken up at the cell body or at the axonal compartment? And therefore we employed microfluidic chambers where you can seed the cells on one side and then the axons grow through the micro channels into the other compartment. And the two compartments are separated. So you can add exosomes either to the soma side or to the axonal side. And even if we added PKH67 labeled exosomes or CRE labeled exosomes, we could see that either to the soma side or also the axon side, and that is the important thing. So we can add the exosomes to the axonal side and see then the signal in the soma, so in the recombination of the neurons. So these EVs can be taken up by the somas as well as at the axonal sites. Then the question was whether neurons somehow may benefit from these EVs, and therefore we performed some assays measuring the metabolic activity of primary neurons and performed stress with these neurons. Under unstressed conditions, addition of EVs or exosomes did not have much of an impact. But if we stressed the neurons and the neurons were treated before the stress with EVs, then we could see a clear effect of the exosomes, meaning these neurons were protected from these stress conditions. So this, if we added the exosomes after the stress, then there was no effect. So important was to treat, in particular in the oxidative stress paradigm, to treat before. And in the nutrient deprivation paradigm, we treat during the nutrient deprivation. And this effect is actually associated with pro-survival signaling. So neurons treated with exosomes activate pro-survival signal pathways like ACT, GSK3 beta, or the Jun kinase pathway. To summarize this so far is, and here you can see again these MVEs present in the periaxonal space close to the axon, and here a cartoon of this whole situation. When the axons are electrically active, they release glutamate, and it is actually known that glutamate can be released along the axon. It's not only released at the synapse, also along the axon. Then it finds NMDA receptors that are present in the periaxonal membrane of the glial cells, which allows the entry of calcium, resulting in the release of multivesicular endosomes and exosomes, which are then taken up again by the axon, or also at the somatic site, of course, and the cargo can be retrieved by the neuron. And the neuronal benefits of that is that the neurons, they acquire a stress resistant stress resistance. I didn't show you some data that we got that they increase their firing rate, which also could reflect that they are more fit and they are more resistant to stress from outside. And they activate pro-survival signaling. And also we have some data on gene expression, which I don't go into the details today. So in total, we think that this is an important mechanism of support and homeostasis, which we further then follow up in our in vivo settings. However, the first main questions that we ask, can we observe this glia to neuron EV or exosome transfer in vivo? And where in the brain or when does it occur? So performed spatiotemporal analysis. And then in the second part, I will also address in vivo models where we can study the function of these EVs. But so in the first part, we're just looking at, can we validate that this EV transfer occurs? And we turned back to the Cree system actually, but modified, and I call it an improved Cree system by using the ERT2 reporter mice. So how does it work? So in the naive, meaning the tamoxifen untreated situation, so first of all, of course, we drive Cree under a promoter that is cell type specific, and in our case, oligodendrocyte specific, which would be the PLP promoter in our hands. So Cree, as I said, under the naive conditions, meaning without tamoxifen, would then be sitting in the cytoplasm and not translocated to the nucleus because it's stabilized by HSP90. And this, according to our idea, would result in sorting to exosome and then transfer to the axon. However, to induce a recombination, animals have to be treated with tamoxifen, and then up on tamoxifen induction, neurons that carry the Cree or that have received the Cree via exosomes would also recombine. So we would see recombination, of course, of oligodendrocytes, but also in addition of neurons. And this could be an indication that Cree was transferred to the neurons. So indeed, this shows that the Cree translocation to the nucleus is highly efficient upon tamoxifen treatment. And we also looked whether we can find Cree ERT2 in the exosome and whether this is dependent on tamoxifen. And indeed, it is. In absence of tamoxifen, we can detect it in an exosomal preparation. And it's almost absent when we treat the cells with tamoxifen. So then this is the quantification of this situation. So it seems indeed Cree under naive conditions is sorted to this compartment and hopefully in vivo also then transferred to neurons, which would result in recombined neurons. So what we then did, we analyzed the Cree ERT2, so the PLP Cree ERT reporter mice, where we have Cree ERT2 expression in oligodendrocytes, and also present TD tomato reporter in all cells, which would then result in recombination of these reporters in all cells where Cree is present, meaning in all oligodendrocytes. You can see the white meta area here in red. And maybe also, if we are lucky, we will find neurons. And these are stained in green here using the marker NOI-N. And if we zoom into here, we indeed can see yellow areas where we can observe co-localization. And then we performed an automated analysis. We first divided the brain into subregions and then scanned these regions for co-localizations, did a circularity analysis. So this was largely a binary analysis to then count the recombined neurons. And another important point is that we had 3 time points of analysis where we played with the tamoxifen injections. So the early time point, we only had 1 tamoxifen injection, and then we had accumulated cycles of tamoxifen and naive wipe out times, where we have then transfer and then recombination, and then again transfer and recombination. And ideally, with more tamoxifen injections, we would think that we have more transfer of Cre and then a larger number of recombined neurons. So we think this could be an indication that it's indeed exosome transfer and not something like we would have to address, like promoter leakiness, which would rather happen in the early phase of development. So of course, we also performed confocal analysis to really show that these are co-localizations. And now let's go into the quantitative data. So here you can see the total brain counts, and indeed you can see between 1 and 12 tamoxifen injections, we can see an increase of the number of recombined neurons in the brain. We can also localize them to brain areas, and we can find them in the deeper layers of the cortex and in the striatal amygdala area largely, also shown here. But in all areas we looked at, we can find this increase with the number of tamoxifen injections, which we think is a good hint that indeed we look at exosome or EV transfer. However, we never can be 100% sure, and therefore we crossed in an EV ablation model. So we used floxed ALIX mice to delete ALIX, specifically also in oligodendrocytes. And in this case, we would anticipate that EV release is ablated, or at least strongly reduced. And then we should not find anymore the recombined neurons, or at least in lower frequency. Here we looked at the knockdown efficiency. With these tamoxifen injections, you never have 100% recombination, and at least in cultured cells, we find about a 50% recombination efficiency. And then we also looked into the particles or the EVs released by cultured oligodendrocytes, and indeed we see a reduction of EV release after tamoxifen treatment of the cells. And if we do a marker analysis, all the markers are reduced by about 50% or even more. So it seems the model works quite well. And in addition, we also performed multivesicular body analysis in situ and could find that upon tamoxifen treatment, the number of multivesicular bodies in the myelinating axon myelin figures is largely reduced. So the knockout model really seems to work quite well. Now let's have a look at the data, and there we were really surprised to see that it worked in such an efficient way. Here, the data that you have seen before, this increase over time, which is now totally gone. Yeah? So we see a kind of the basal recombination, which we actually would expect because the knockout has to penetrate first, and then largely the increase of the neuronal recombination is completely gone in all of the brain areas we looked at. So now we are actually quite confident that with this model, we really can display and prove that EV transfer from the oligodendrocytes to the neurons indeed occurs. And now I hope you are quite the same way convinced as we are. So let's come to the functional role of these exosomes that are transferred to neurons. And I would like to come back to these two knockout models that I had introduced before, the CNP and the PLP knockout. As I told you, we observe in these knockout models an axonal degeneration. And so we ask whether these knockout models something have to do with exosome transfer, and whether exosome transfer may be important for long-term axonal maintenance. And there had been some other studies before showing that glial support that was associated with these two models is due to transfer of energy-rich metabolites from oligodendrocytes to neurons. So glycolytic substrates like lactate or pyruvate, which are transported through MCT transporters to the neurons, and there provide the neurons with energy. So far, so good. There are quite substantial data on this, what is called metabolic support. However, it is totally unclear at the minute how this is molecularly related to the PLP or CNP deficiency. So this is where we asked, has it something to do maybe in these two mouse models, at least also with EV transfer, because we find PLP and CNP both associated with exosomes from wild-type oligodendrocytes. So we analyzed exosomes or SEVs derived from PLP and CNP knockouts and compared it to wild-type animals. And indeed, the number of EVs, and this is now normalized to the number of secreting cells, is reduced by about half in both knockout models. And all the markers that we looked at and quantified are clearly reduced. So we have a quantitative defect of exosome release. And we also looked at the multivesicular bodies, and there we also can find some interesting data, and they differ between the CNP and the PLP knockout a little. So in PLP knockout, we have an accumulation of these MVBs. Yeah? So the number in CNP is normal, and in PLP knockout, we have an accumulation. And this accumulation, when we look at their place, whether they are at axonal, so directly close to the axon, within the myelin sheath, or abaxonal, or maybe in the cell bodies. So we quantified this independently, and they are largely enriched in the places away from the site where we think the release occurs. So it's a kind of a build up, an accumulation of these EVs, so probably of these MVBs, probably they cannot be released for some reason. So in the CNP knockout, it's slightly different. We have a normal number, but we have a redistribution of these MVBs. It seems they cannot be transported to the adaxonal site. So they are reduced at the release site. So we have also morphological evidence that EV release in these two knockout models is indeed impaired. Then we performed quantitative proteomics and compared wild type and the CNP knockout EVs. And surprisingly, they shared quite many common features, even in the upregulated, but I think, at least to me, more interesting, the overlap in the downregulated proteins, because 6 out of the 10 most prominently down-regulated proteins in both PLP and CNP knockout were actually identical. That these are the red ones here shown for the PLP and the CNP knockout. And they are now indeed real good candidates that we can look at, and that they may have a functional role in these EVs and in the potential support function. So we've validated these data also by semi-quantitative Western blotting, and could find actually also a reduction of ALIX that we could not find in the proteomics, but most of the markers we actually could validate it. Of course, well, these Western blotted quantitations very often are very variable. But we are happy to see that largely we could validate what we also had seen in the proteomics. So, but this means on top of the quantitative defect that we see, we have a qualitative abnormality of these exosomes, because here they are normalized to particle number and here normalized to total protein in the proteomics, of course. Then we looked into the function, and I had introduced you to the support assays, the MTT assays that we do with the primary neurons, and then we applied wild type and compared to the knockout exosomes to the neurons and performed the MTT assay. And first of all, you can see that the support function is, there is a dose response curve. So at about 50% of EV reduction, we actually already lose this support function. And this is what we actually would expect, and that we indeed see when we normalize the EVs that are applied to the neurons according to the number of EV secreting cells. So this is the support function of the wild type. We lose it when we do a 100,000 G depletion. And on the very same level are the two knockout EVs when they are normalized to the EV secreting cells. But then the exciting question is it also abnormal, or do we lose the function also when we normalize to the SEV particles? And yes, indeed, we also lose the function. So again, also on the functional level, we have a qualitative and a quantitative, here the qualitative and here the quantitative defect that we can observe. We further looked into axonal transport, because axonal transport is a very important feature of axons that is required to keep the axon healthy. And when axons are degenerating, one of the first hints that you can see that the axon indeed is starting a degenerative process is that axonal transport is impaired. So we did in vitro live imaging of cultured hippocampal neurons. These were transfected with a BDNF M-cherry construct, labeling vesicles that are traveling up and down the axon. You can see the axon here, and all the vesicles that are traveling up and down the axons, which then can be quantified by live imaging. You do some chymographs, and in these chymographs you can then follow the traces and then quantify all different types of parameters. And what we saw when we treat the neurons with normal wild type exosomes under unstressed conditions, we see actually a reduction in the pausing time. So the axonal transport usually is driven, and then there are pauses, and then it's driven again. And the time of the pauses, the relative time of the pauses was reduced when exosomes were added, leading to an overall faster transport of the cargo. However, all the other parameters were not changed when we added the wild type or the exosomes under non-stress conditions. So we moved to the nutrient deprivation conditions and treated the neurons with SEVs overnight, and during the last 3 hours they were subjected to nutrient deprivation, followed by live imaging. And then we compared the nutrient deprivation situation, which means we applied 100K supernatants carrying all other soluble factors that might have an impact on axonal transport, but lacking EVs. And added then 10K supernatant, which carried the EVs together with these other factors that might be present. But we had the appropriate, as control, we used the 100K depleted supernatants. And as you can see, in the nutrient deprivation, and you can also see the videos down there. In the nutrient deprived situation, the vesicles move less frequently. And if we add the wild type SEVs, this is kind of restored. Here you can see the chymographs also showing this. And if we add the knockout EVs, and here we actually added also astrocyte-derived EVs, which I initially thought would be not a good control because I expected them to have the same effect as the oligodendrocyte-derived EVs, but now we find it's a perfect control because these EVs do not show the same effects. We performed a quantification, and what looks quite complicated is not so bad to read. If you focus on the red bars here, you can see here you can see the nutrient deprivation effect, and the red bars you can see it restored to the level of the gray bars all the time. And while the knockout and as well as the astrocytes, they are more on the level of the nutrient deprived supernatant. On the 100K supernatant. So we lack this axonal transport promoting activity of the wild type EVs in these knockout EVs. And this is actually true for all the parameters like anterograde trafficking, retrograde trafficking, the number of static vesicles, and the pausing time of these vesicles. So this means that we lose the axonal transport promoting activity of the wild type exosomes in these PLP and CNP knockout animals. So to summarize this, I have shown you that these oligodendroglia derived exosomes promote neural stress tolerance, and a major part of this obviously seems to be that they have the ability to promote axonal transport. This provides a kind of a neuroprotection. We call this glial support that is provided by the oligodendrocyte to the axon. And we think that this is in long term very important to maintain axons over a whole life. And if we lose this function, as we could see in the PLP and the CNP knockouts, which actually develop this axonal, this secondary axonal degeneration, then we lose the exosome functions and we lose the axonal maintenance function. So, and we also use discrete ERT reporter models, and in addition, on top of that, the conditional deletion of the exosome release. I didn't talk about RAP35 knockouts. We also have these animals at hand, and it seems also that they have a reduction of about 50% of EV release. But in this case, in the CREAT2 models, we only use the ALEX knockouts right now. And these really demonstrate or provide, I think, quite convincing evidence now that there indeed is clear to neuron exosome transfer in the normal mouse brain, in the unmanipulated animal. So to bring this maybe into a picture that is more easier to understand, there is something like an M&M support in place for neurons, for myelinated neurons. So there is substantial data on the metabolic support, which says that oligodendrocytes provide energy-rich substrates, fueling the axons, and which will satisfy the high energy demands that axons actually have. But I think we have now shown that exosomes provide a kind of a molecular support. And then, of course, now we are interested what these molecules are and what are the molecular mechanisms that are in place that help these neurons then really to maintain their architecture and to keep the axonal transport in place. So there is a substantial work, of course, still to do. And as I said, you should keep in mind that these neurons, they really provide, they have very, very long axons, and the biosynthetic machinery is actually in the soma, and all the things have to travel down the axon, which takes months or sometimes even longer in a human who has axons of sometimes more than a meter in length in the corticospinal tract, for example. So we think it's quite logic on some hand that the glial cells that sit next to the axons provide their biosynthetic machinery maybe, and provide the axons with molecular support that is important for these axons. So now at the end, in the last few minutes, I would like to share with you also some of our pain that we had when we tried to analyze microRNAs associated with oligodendroglial EVs. And I term this fact and artifact, this part. So of course, it's a very attractive hypothesis that small RNAs or microRNAs are delivered via exosomes or EVs from glial cells to neurons and modulate the neuronal functions. So, and of course, we also followed this hypothesis. And when we looked at the RNA that we can recover from our exosome preparations, we can see the typical small RNA peak of RNAs below the size of 200 nanometers. So we performed a small RNA sequencing and found what basically everyone sees. There are specific microRNAs that are highly enriched in the exosomes versus the cells, and this could be our candidates that we follow up. And then we performed a second RNA-seq where we had treated neurons with exosomes and sequenced the neuronal RNAs to find potential target genes. So there, and we found some that are down-regulated, others were up-regulated. And then we thought the initial idea was that we bring these two data sets together, and then we find the microRNAs that regulate specific genes with defined functions, let's in axon biology or maybe in neuronal plasticity. So what initially motivated us a lot, and as I show you here, we had two candidates that were of main interest and that from their biology would fit very well in the metabolic support function of glia that glia might have. So we looked at these microRNAs that are enriched in exosomes, and we could validate this in other exosomal preps, cells versus exosomes, again upregulated, while other microRNAs stayed constant. But then at some point we looked also at EVs that were cultured in the absence, oh, I forgot. And we thought we are in a very good shape because we always culture our cells under serum-free conditions. And so we use media supplements like B27 or NS21, which is a specific supplement for oligodendrocytes. So we then, at some point, I was suspicious about this strong enrichment here. And then I said, we need to do this control. And indeed, after collecting EVs in the absence of the supplement, we could not detect these two microRNAs anymore. Then we analyzed the medium only, and indeed we could find it in the medium only, all the microRNAs, and only those that medium, the basal medium was microRNA free, but it was actually the supplement that carried all these microRNAs into our qPCRs and probably also into our RNA-seq. So really a major background which we never had expected, and so people really have to take care even under serum-free conditions and make always appropriate controls. So that's what we actually did in the next experiment. We did a follow-up sequencing where we compared EVs that were collected with the supplement or only the supplement, and also generate a sample where we only sequenced the media that was subjected to the EV preparation procedure, of course. And then we can see in the NS21, in the supplement only, we can see an enrichment of several microRNAs, which are actually also present in the sample plus the supplement. And we could see that it's actually 90% of the microRNAs found in the media that make a substantial part of the whole EV sequencing results. But what we then now propose is that we simply subtract these microRNAs, not the number of reads, but take them completely out of the sequencing data and not considering at all, because simple subtraction of the reads does actually not work, because we do not have a real normalization that we can do there. So that's what we actually now propose, that we have to do this media control sequencing and simply correct the data sets by these microRNAs that are found in the supplement. So this is where I would like to stop and thank the people that were involved in the work. There is a group of people that have done the initial work: Carsten Fröbeis, Dominik Fröhlich, Ping, Ko Elsner. They have done the basic tissue culture analysis of EV transfer from oligodendrocyte to neurons. And this was taken over by Kerstin Miebach and Tini Müller, who did the CRE-ERT2 work. Martin did the microRNA analysis. And Alla actually is working on EVs that are released during exercise, which could actually be another topic during this WebEV series at some point, if you are interested. And of course, we are thanking all our collaborators, which are very important for us. They provide us with all the transgenic mice and also important techniques like Letizia Peris. She taught us on the axonal transport assay. And of course, we thank all our funding sources. And I would not stop before acknowledging also GSEF. Please check out the Extracellular Vesicles DE site about the activity of the German Society of Extracellular Vesicles. We are having meetings together with other European societies. We hope that we can have these EV mobility fellowships soon, after when Corona will allow. And also check out the recent edition of Trillium Extracellular Vesicles. This is the second issue this year, and there are some really interesting articles, everything freely available online. So thank you very much for your attendance, and I'm happy to take questions.
Thank you very much. So please state your questions in the chat and I will let you unmute. And I think we'll start with Clotilde and then we come back to Carolina a little bit later. Please unmute yourself, Clotilde.
Okay, thank you, Jan. Thank you, Eva. It was really a great talk, and I really admire a lot all the models that you're using. It's really beautiful. I have one comment about tamoxifen, and we discussed that, I think, in one of the
I remember
Barcelona.
Yeah, so, it's a long time ago. Yeah.
So we in the past, we did some CRE-ERT2 inducible mice crossed with RAP27 flux in the hope to induce RAP27 knockout upon tamoxifen induction, and we generated cells from that. And then we tried to knock out RAP27 to inhibit the exosome secretion, etc., with these cells. And we noticed that tamoxifen itself had an effect. I don't remember now if it was increasing or decreasing or whatever, but it had an effect on the EV release. So I think it does not impact your results at all because you're looking at recombination. So I think it probably does not impact what you've looked at. But if you wanted to use these models for validation of the in vivo function of the EV transfer, then you have to keep in mind that maybe tamoxifen itself could have an effect. I wonder if you have any info on that?
So you're completely right. And I often get these questions. There are different scenarios. I think your idea is that it has an impact on EV release. Other people think that it might have an impact on reporter gene recombination. You know, that if you inject with tamoxifen, you induce a reporter gene recombination. So,
right.
And you alerted me a lot when you talked to me in Barcelona, and we actually had a look whether, at least in vitro, whether tamoxifen affects EV release from oligodendrocytes, and it doesn't.
Mm-hmm.
So.
Okay.
So this is clear, and we also did, of course, controls of injecting reporter animals that do not carry the CRE, yeah, only the reporter, with tamoxifen, that we do not have a leakiness of the reporter model there. So we did several controls, and I think that we are actually in a quite good shape. So that's why these studies take such a long time. And I think now we are almost there. We still collect some data. So of course, yeah, we really, yeah.
Thank you.
Yeah.
No, that's great that you could make these controls and that they work.
Yeah,
and
we will see what the review. We will see what the reviewers say.
Yeah.
Probably they find something else.
From
So if I may have a quick question, is it okay, Jan?
Yes.
Okay, so the quick question is about the oligo-derived EVs from the PLP knockout or CNP knockouts that you showed that they had a trophic effect on the neurons at the end. Something I may have missed, but in your original scheme, EV release by oligo was induced by glutamate signaling by neurons, right? And yeah. in this in vitro assays of the trophic support, did you induce EV release by glutamate or anything, or was it the spontaneous release? So that if not, then maybe in vivo, the situation would not be exactly the same. Yeah, okay.
Yes, indeed. So all our functional assays, we conduct them under, let's say, basic release conditions. We do not treat the cells with glutamate. I think we tried at some points and whether there is a difference, and we couldn't see a difference, but the glutamate treatment induces, it makes the data more variable. So therefore we stopped the glutamate. Yeah, we did not. But you're right, in vivo, we are in a different situation. And actually we tried to mimic the electrical activity the electrical activity in the in vivo model, meaning, so we tried to trigger or remove certain electrical activities, and then we should see a difference in the neuronal recombination. You know, so actually this was not a very nice model. We removed one eye of the mouse, and then you can compare the neuronal recombination in the visual cortex. Yeah, and you should see then recombination on one side and not on the other side. But unfortunately, you know, we did not cut so we missed the site where we actually would have found the recombinations, and so we would have to redo all the work. So this was a bit of disappointment. So it's not so easy to really just look into the brain and count the recombinations, but that was one idea we had initially, and which I found quite important, and unfortunately we lack this connection to the neural activity now, which would have been a nice addition.
Well, thank you. It was
Carolina,
great work.
you had a question?
After
that, I have a question myself.
Yeah, I think I have a quite naive questions actually. I was just wondering whether we can categorize synaptic vesicles as extracellular vesicles, because by definition is vesicles being secreted as well. What do you think, Eva?
Yeah, no, I really would. This is really something absolutely different, because I mean, a synaptic vesicle is a cytoplasmic vesicle it has the opposite topology as compared to, so inside the vesicle is actually topologically equivalent to outside of the cell. And so it's the content that is released, yeah, and not the whole vesicle. So maybe you could call it, it's an inverted extracellular vesicle that is inside the cell. No, so it's an intracellular vesicle and not an extracellular vesical.
Yeah, so it basically delivers us the transmission factors, right?
Exactly.
But then, well, ages ago, I was doing my PhD. I learned a little bit about it, and there is some sort of similarities in terms of molecules that are involved in the synaptic vesicles, endocytosis and exocytosis. So if you play around with the extracellular vesicles exocytosis, will that also influence the synaptic vesicle release? So such as like a snare proteins, you know, there's some similarities in it.
Yeah. I did not think about the snare proteins. I mean, the funny thing is, and I don't know if Clotilde knows a bit more about it by now, it's not known a lot about snare proteins regarding extracellular vesicle release, which of them control EV release. It's only a few a few studies that have data on that, and I think several people have looked into that. But anyway, I think the common scheme may be that synaptic vesicles are also generated through an endocytic recycling machinery. And there might be some factors that are shared, maybe like GTPases, or that regulate this endocytic, the endocytic function. But of course, at some point, the synaptic vesicles, they recycle back to the plasma membrane, while the EV biosynthetic or the exosome biosynthetic pathway would separate from the early endosome towards the late endosome. And there are other factors, and in particular, when the vesicles, the intraluminal vesicles are generated, I think the machinery is completely different as compared to the synaptic vesicle regeneration machinery. So maybe at the level of Rep5, there might be some similarity, meaning the endosomal, the early endosomal pathway. But as soon as the EV biogenesis enters the late endosomal pathway, I think it's a different, that the two pathways dissect.
Okay, we'll allow two questions, and one of them is my own, but we have Hernando. Please unmute yourself, yeah.
Thank you, Jan. Thank you, Eva, for such an excellent talk. This is maybe a naive question, but I would like to know what is the norm of the exosome communication from the axon to the oligodendrocyte? Is there any benefit? Is there anything about that is known about that, Eva?
You mean in the other direction?
Yes, I mean, because I guess, you know, they also release, you know, messages. So somehow I'm looking into what is the benefit, you know, is there any mutual communication or is only unidirectional?
So I think what we can exclude is, so if we define an axon as the long, thin process that projects away from the cell soma, so between the soma and the synapse, I think we can exclude exosome release from the axon, because there are I think no one has observed multivesicular bodies there. There are late endosomes that are travel back to the soma, but so I think multivesicular bodies are largely in the somatodendritic compartment, and there indeed definitely is exosome release from the somatodendritic compartment, but they not really address oligodendrocytes, and I think they are not taken up and the cargo is not retrieved. Maybe they have a signaling function. Yeah. I think Remy Sadoul looked at this at some point, and he could find that the vesicles, they bind to the surface of the oligodendrocytes, but they are not internalized, or only a few of them, or not efficiently internalized. But it seems there is not a prominent pathway from the neuron to the oligodendrocyte, not to my knowledge. And we not really have looked into that, actually, I have to say.
Clearly suggesting maybe budding from the surface of the axons. Does that happen?
Yeah, and from the axonal surface.
Yeah.
Yeah, I wouldn't exclude that. Yeah.
But it's not
I think there is some
big product.
So the early electron microscopists, they actually find some strange figures where the oligodendrocytes somehow, maybe you would call that togocytosis, picks up some axonal membrane, or the axonal membrane picks up some oligodendrocyte membrane. So there are some processes that we actually also have to think about when we look in our in vivo, in our CRE-ERT2 animals. So I think we are in a quite good position because we also have the ALIX knockout. But I mean, yeah. So I wouldn't completely exclude that the neuronal, although I'm really convinced that we look at exosome secretion, all the data that we have point at exosome secretion, I wouldn't completely exclude that there are also other mechanisms of exchange between neurons.
So there's some effort of isolating extracellular vesicles from different tissues. We've been working on tumor tissues, for example, in my lab, and others have worked on brain. Andy Hill and others have worked on brain tissue extracellular vesicles and isolating them from small pieces of brain. So firstly, what's your view on isolating vesicles from brain tissue, and what is the relative? What do you think is a relative contribution of different cells to that vesicular pool? I think, I mean, the next field of research, the next horizon we have to reach is to understand EVs in tissues, and you're trying to do that, and so are we. So, but comment on the general brain EV, please.
So my opinion is, and you can see this from my research, that we need to use transgenic animals to study EVs in the brain. So I'm quite skeptic, I have to say, regarding the isolation of EVs from the brain. I mean, the brain is really a very tight tissue, so it's really cells sitting next to each other. Everything is quite compact. So when you homogenize a brain, you will definitely generate homogenization. I call it artifacts.
Well, it depends on how you do the process. I totally agree. If you whip the tissue, you will break up cells like crazy. But if you do it with some care,
you can do an enzyme digestion, right?
Yeah.
But anyway, I mean, cells will die. You will generate fragments that are present, and that will come into the EV prep. I would be surprised if that would not happen. So I would, yeah. So what I think the strength of these methods is in comparing different situations, like different disease states or different knockout animals, for example. There, I think there is definitely a use to it, but I always would be skeptical regarding whether yeah, I think it can be argument within other arguments, within other experiments that you perform, but as a sole argument, these are EVs coming from the brain, and this is what I'm characterizing, and they do this or that. I would be skeptical.
There's still more technology development that's needed. I totally agree with that. One question on your there's your supplement you were using that contained microRNAs, obviously, right?
Yeah.
And you don't know exactly what the supplement contains, is that correct?
No, we know it, correct. So we know it. We have the complete list of ingredients. And what I didn't tell you, we checked any single ingredient, yeah, of the which one, and it's indeed there is catalase in. and it's the catalase, and it's only the catalase, and you can order so we were trying to make our own supplement so that we can supplement our cells with a microRNA free medium.
Yes.
But we couldn't, because we couldn't order, we couldn't get a catalase from any supplier that was free of microRNAs. So this is quite interesting, and I was trying to.
So how this is produced by cells, right?
Catalase.
How
so the catalase that is distributed, this is isolated from liver or from blood.
Right. So my question is, my big question is the supplement contained in exosomes?
How could it contain exosomes? I mean, the supplement is
if
produced.
It contains microRNA, where are the microRNAs unless they are in vesicles?
So I think they bind to a protein. So this is, so if we
anyway,
buy,
I asked a difficult question.
No, it's, I don't think it's difficult. It's fine. Yeah. It's fine. Right. We buy a 99% pure catalase. Oh yeah, and this contains microRNA. So I cannot imagine that. Yeah. So we haven't put the catalase into the NTA machine, I have to say. But I think the more easier, let's say, explanation is that catalase itself binds the microRNA. It has positive charges. So I think maybe there's a biology. I would rather think that there is a biology to that catalase is binding microRNAs, than thinking that EVs.
Thank you, Eva Maria, and I'll give the word to Carolina to follow up and invite you all to the next series of events. Thank you.
Welcome.
Yeah, thanks so much, Eva Maria. It was really wonderful talk. Lots of information. I think we learned about how the EVs from oligodendrocyte providing support for homeostasis in the brain. Just to put it very shortly, out of like so much data and fantastic results. So thanks so much for that.